The other trial I need to talk about here today is another trial that series of trials that came out of Dr. McKay's lab as well. This one is focused on white mold. So just as a quick refresher, I think most of us in northwest Minnesota and the Red River Valley are all too familiar with white mold. But just as a reminder, this is a pathogen. It's long-lived in the soil, produces survival structures that can live for about 5 years or more. And when we have wet weather, cool weather, particularly during flowering, we can see disease. And it can cause very significant yield losses. So one thing, and one of the reasons why we deal with it here, is that we have a lot of very susceptible crops in our rotation. So pretty much most of the broadleaf crops that we grow in the Red River Valley and beyond are susceptible to the pathogen that causes white mold. Like I said, it can persist for a long time in the growing or in the field. And so those two combinations, the large host range, the long-lived survival structures, mean that we can't rotate our way out of this particular problem. We do have varietal resistance. It's not complete by any means and is oftentimes a little bit less than what we would hope for. Chemical management is one option. Timing is critical, as is coverage down within the canopy. And if you have a year that's particularly favorable for white mold, you're going to get some yield loss, even with a fungicide application. Okay, so they had two different trials. I have time to talk about one today. Basically, the idea is to figure out ways to better our resistant varieties here in the state of Minnesota. So these trials are in collaboration with Dr. Lorenz's lab, so the University of Minnesota soybean breeder. And basically, they want to figure out which traits related to soybean plant architecture can help the plant to evade or escape infection. So we have something called physiological resistance, which is when the pathogen actually tries to get into the plant is able to resist it. The plant architecture traits are an escape mechanism. So the thought is that a bushier soybean variety doesn't allow as much airflow. It doesn't. It allows underneath the canopy to be a lot more shaded, so a more favorable environment for white mold to occur. A more upright plant architecture is the opposite. The theory is. So what they did is they looked at all kinds of things. So there was a series of 150 breeding lines that they screened. They screened it for all different types of traits. Again, relating to how much light the plant was, that particular line was able to allow to reach the soil surface at various distances from the main stem. They looked at, so this is canopy closure, so weekly they went out and figured out, okay, all of these different colors are different soybean varieties. So each week they wanted to take a look at when canopy closed. So what they did is they flew a drone over these fields to figure out when canopy closure occurred. They looked at petiole slope. So I wrote this down here because let's see. So the petiole slope Okay. Plants with a lower slope have a more rounded canopy and are thought to allow less light through. So this is the pale slope. They looked at average leaflet area. So again, each of these different colors is a different soybean line. And then they looked at the ratio of leaflet length to width. This is basically a measurement of shape. So more compact leaves would allow more light to penetrate that canopy. So what they did out of those 150 soybean lines, they narrowed it down to 20 lines that they were going to use for further study. And they selected those based on canopy closure at this July 21st date. They have a large range there. They have low canopy closure lines, medium, and high canopy closure lines on that particular date. That date is significant because that's when flowering began. And as I might not have mentioned, but that's how the pathogen is typically able to infect the plant. Spores land on senescing soybean flowers and use that as a food source to get into the plant. So then they went out into the field and took a look at these 20 lines in a little bit more detail. So one of the things they were looking for were these little cup-shaped mushrooms. So the pathogen is called Sclerotinia sclerotiorum. Sclerotinia sclerotiorum. That particular pathogen produces those little cup-shaped mushrooms. So they wanted to see where they saw these apothecia underneath individual soybean lines. She only found one. This was the work of Alicia Hirschman. And she was excited to find this one, but the plant was at a later reproductive stage. They also looked for disease incidence and severity, and that's when you combine those together, that's our disease severity index. Each of these different breeding lines here, they looked at for disease incidence and severity. So a disease DSI of 0 means there was no disease, and if you see a number in there, there was some disease. So they had a very upright check, and they had a bushy check. The bushy check did have disease, the upright check did not. The thing that's significant there is that those particular lines don't have that physiological resistance that I talked about. The reason why that's important is because if you can find soybean lines that have both physiological resistance and these avoidance mechanisms, you might be able to breed a soybean variety with just a better resistance profile overall. So they are actually looking at all of these 20 lines in the greenhouse, so they're actually physically inoculating plants, and this is to look at that physiological resistance. Like I said, they're going to take these lines again outside in 2024. The reason for this is they want to make sure that these plant architecture traits are something that is conserved in that particular line and isn't something that is just heavily regulated by environment. Okay. So this other young man, young man, he's looking at different populations of the pathogen, and he's developing a panel that can be used to screen resistance or for resistance in the soybean germplasm. I'm not going to show you this. This is what I wanted to show you. So here's a lot of the people in Dr. McKay in her lab. There's a picture of Dr. Lorenz here because these two projects go hand in hand with his breeding program.